Development of PCR protocol for detection of Escherichia coli in drinking water. The outline or flow of the methods are the following: reagent preparation, followed by extraction of genomic DNA, PCR optimization, sensitivity of PCR, protocol for sample analysis, and lastly, the analysis of result. To understand why this study is conducted, the background and objective of the study should be considered.
So, before we proceed to the methods that are involved in the study, let us first give you a background of this journal article. In developing countries, the major cause of sickness in humans are water related diseases due to the consumption of untreated and contaminated water. In Pakistan, which is the setting of this study, the consumption of contaminated water is a common occurrence due to the deficit in drinking water treatment facilities and mismanagement of the existing ones. The diseases presented here, such as hemorrhagic colitis, hemolytic uremic syndrome, and thrombotic thrombocytopenic purpura, are associated with Escherichia coli O157, H7, and enterohemorrhagic Escherichia coli strains, which produce Shiga toxins. The major reservoir of Escherichia coli that has these potent cytotoxins are the environmental waters. The methods to be presented here may facilitate epidemiological surveillance of water and edible products for human consumption to determine the risk of acute diarrheal disease at any single time in any densely populated geographical region.
The methods currently used by some laboratories to test the bacteriological contamination of drinking water take about 2 to 4 days, and the presence of non-culturable strains can sometimes yield inaccurate results. Polymerase chain reaction or PCR may facilitate epidemiological surveillance of water and edible products for human consumption to determine the risk of acute diarrheal disease at any single time in any densely populated geographical region. However, the sensitivity of PCR can be influenced by physical dilutions of the samples, and this can be substantiated by coupling PCR with the enrichment step. Enrichment can lower the detection limit and dilute the inhibitory substances present in the sample and may also reduce the possibility of detecting dead cells. The objective of the study is to develop a PCR protocol for the detection of E. coli O157H7 and E. coli virulence gene SLT or Shiga-like toxin I in drinking water using a double enrichment step in order to monitor the microbial quality of drinking water within 24 hours.
For reagent preparation or the materials used, in bacterial strains and culture media, the pure culture of Escherichia coli O157H7 strain EDL 933 is obtained from the American Type Culture Collection Center. Strains were nutrient agar slants and incubated at 37 degrees Celsius. Primer pairs are used for E. coli detection. These are RFB, F, RFB, R, SLTIF, and SLTIR. This set of primers helps in revealing the serotype O157 of E. coli and also its virulence trait Shiga like toxin production. These would be helpful in serotype identification and toxin profiling of the species prevalent in the region. DNA ladders or NORGEN of 50 and 100 BP or base pairs have been used for comparing the extracted DNA of the microbe and to determine the PCR product size. This table shows the target genes of Escherichia coli and their primer sequence details. target gene 0157, the product size is 292, while in SLTI, the product size or the base pairs is 210. In template preparation with two methods which are phenylchloroform method and genomic DNA isolation kit, the reagents used are 200 microliter of lysis buffer pH 7 to 8, 55 microliter of 5M sodium chloride solution, chloroform, ethanol, 50 microliter of 1-XTE buffer, 30 milligrams of wet bacterial pellet, 0.24 ml of homogenization buffer, 0.8 ml of protein precipitation buffer, 0.62 ml of isopropanol, magnesium chloride, 10x PCR buffer, 5-U-Tac DNA polymerases, 1 mm magnesium chloride, ethidium bromide, and 1x TBE buffer.
For the extraction of genomic DNA, the methodology used was phenylchloroform method and genomic DNA isolation kit. For the genomic DNA isolation kit, the researcher used PrepEase Genomic DNA Isolation Kit, Affymetrix product number 78850, which can be prepared for 50 preps and 250 preps. The kit provides a simple and efficient procedure for rapid isolation of genomic DNA from various samples, which includes mouse tails, animal tissues, cultured cell, bacteria, which is used in the research, the yeast, wool blood, and plant leaves. The procedure does not use phenol or columns and can process a large number of samples in a short time. For the genomic DNA isolation kit, the procedure are as follows. First, the fresh culture of 300 milligrams wet bacterial pellet was used for extraction of DNA. The sample was sliced by adding 0.24 milliliters of homogenization buffer and mixed well on a vortex mixer. A 0.2 ml of chloroform or isoamyl alcohol in a 24:1 ratio and 0.8 ml of protein precipitation buffer was added into the lysate. After that, the mixture was vortex, followed by centrifugation at 13,000 rpm for 4 minutes. After centrifugation, 0.88 ml of supernatant was transferred into a 1.5 ml Eppendorf containing 0.62 ml of isopropanol. Then the genomic DNA precipitate as white, fiber-like strings. The solution was mixed thoroughly and centrifuged at 13,000 revolutions per minute for 4 minutes. After centrifugation, DNA pellet was obtained and supernatant was discarded. The pellet was washed with 1.5 ml 70% ethanol by vortexing, followed by centrifugation again at 13,000 revolutions per minute for 2 minutes. Then the supernatant was aspirated without disturbing the DNA pellet. For the last step, the pellet was dried for 5 minutes at 37 degrees Celsius and resuspended into 150 microliter of DNA resuspension buffer. It is vortexed to dissolve the DNA pellet and store at 4 degrees Celsius.
In phenylchloroform method, the 1 ml suspension of bacterial culture was centrifuged for 3 minutes at 12,000 rpm. Then the supernatant was discarded and a cell pellet was used. Thereafter, the cell pellet was well mixed with 200 microliters of lysis buffer, which corresponds to 40 millimeter tris acetate, which has a pH of 7.8, a 1 millimeter EDTA, a 20 millimeter sodium acetate, and a 1% SDS. And take note that RNA is added if it is required for removing RNA from a sample. After this step, 55 microliters of 5 NaCl solution was added in order to remove the cell debris and proteins. And then, the mixture was centrifuged again at 12,000 rpm for 10 minutes at 4 degrees Celsius. After centrifugation, the supernatant was transferred into a new vial and equivalent volume of chloroform was added into it. The Eppendorf was inverted 5 to 10 times, which results to a milky solution, and then it is centrifuged again at 12,000 rpm for 3 minutes. Next, the extracted supernatant was used and 100% ethanol is added into the vial. And then the solution will put into 12 degrees freezer for 1 hour, which will make the DNA precipitated. Then the solution again is centrifuged at 12,000 rpm for 5 minutes, and thereafter the supernatant is discarded, and then the DNA pellet was further washed twice with 70% ethanol, followed by centrifugation at 12,000 rpm for 3 minutes. And lastly, it was then dried in incubator at 37 degrees Celsius for 15 minutes. Consequently, the pellet was dissolved into 50 microliters of 1 times TE buffer, which corresponds to a 1 tris HCl, which has a pH of 8, and a 0.25 EDTA, which stores at 20 degrees Celsius.
Polymerase chain reaction was optimized for Escherichia coli genes O157 and SLT1 with pure culture of E. coli. It was optimized by adjusting different parameters such as magnesium chloride concentration, which varied from 1 to 3 millimolar with increment of 0.5 millimolar. Magnesium chloride is very crucial for the activity of Taq polymerase as it acts as a catalyzer of the enzyme reaction. The second one is annealing temperature, which varied between 48 to 61 degrees Celsius by increasing 1 degree Celsius at a time. In this process, lowering the temperature enabled the primers to attach on their target sequences on the single-stranded DNA. And lastly, primer concentration. Primers, which are short sequences of nucleotides complementary to both strands of double helix of the DNA that bind the 16S rDNA region to initiate the replication process. It was checked at 3 points, 20 picomolar, 2 micromolar, and 10 micromolar. Each parameter in the listed order was optimized while other kept constant. The PCR reaction mixture for the reference PCR protocol is consisted of first, 10 times PCR buffer consisting of 50 millimolar potassium chloride, 10 millimolar tris hydrochloride, and 0.1% Triton X. We all know that a buffer is needed to create the optimal chemical condition and keep the mixture at the correct pH for PCR reaction. Next is 5 enzyme unit Taq DNA polymerases, simply known as finzymes. By definition, a Taq polymerase is a thermostable enzyme that is able to recognize the prime template complex that extends the copy of DNA strand by adding nucleotides to the primer. Next is 1 megamolar magnesium chloride, simply known as Biron, 2 micromolar H primer or IDT, 200 micromolar each of deoxynucleoside triphosphate. Deoxynucleoside triphosphate are the one being incorporated into the newly synthesized strand by Taq polymerase. PCR amplification or cycling conditions. It was performed in EG 9600 PCR gradient thermal cycler, also known as Extragene USA, wherein denaturation is done using a 94 degrees Celsius for 2 minutes. Annealing is done with 35 cycles of primer consisted of 55.5 degrees Celsius for RFB, 54.2 degrees Celsius for SLT1, and 72 degrees Celsius each for 30 seconds in each cycle. In extension, it uses 72 degrees Celsius for 5 minutes.
We now go to sensitivity or detection limit of PCR. The PCR sensitivity for the detection of the bacteria, E. coli, in drinking water was determined through the following methods. The spiked water samples of known microbial culture volume is diluted in 10-fold serial dilution and is incubated for 4 hours at 37 degrees Celsius. It is followed by plating the diluted sample in 1 ml on Luria Bertani agar or LBA plates, where the average cell number is counted through the cell suspension in each dilution from 3 replicas. On genomic DNA, it is extracted using phenylchloroform method by taking 1 ml from each suspension. DNA is then used for PCR by ensuing optimized conditions. For analyzation of amplified PCR product using electrophoresis, an aliquot of 10 microliter was loaded on 1% in agarose gel stain with ethidium bromide and is run in 1x TBA or trisborate EDTA buffer at 70 volts per centimeters for 45 minutes. Results of PCR detection is discussed on the succeeding slide.
Okay, for the methods of protocol for sample analysis. So here, in the reading journal, the researchers did was they concentrated a liter of water sample spiked with Escherichia coli O157H7 strain. This is by filtering it through a 0.45 micrometer pore size nitrocellulose membrane. So a nitrocellulose membrane is the most popular matrix used in protein plotting. So and then what the researchers did is the filter was vortex in a nutrient broth and the broth containing the filter incubated at 37 degrees Celsius for 4 hours.
After the broth containing the filter was incubated, it was then centrifuged at 5,000 rpm for 10 minutes to recover the bacteria. The obtained bacterial pellet was subjected to DNA extraction and was used for PCR detection. The primers, Shiga-like toxin 1 or SLT1 and RFB were used for targeting the O157 and SLT1 gene of E. coli.
In Figure A, Lane 1 shows 1 kilobase DNA ladder, while Lane 2 shows O157 gene having 292 base pairs amplified with RFB, F&R primers. O157 gene gives best yield, having a product size of 292 base pairs at 52.5 degrees Celsius with 2 millimolar magnesium chloride and 2 micromolar primer concentration. On the other hand, Figure B having a lane 1 shows 50 base pairs of DNA ladder, while lane 2 shows SLT1 gene having 210 base pairs amplified with SLT1 F and R primers. SLT1 gene gives best yield having a product size of 210 base pairs amplified at 54.2 degrees Celsius with 2 millimolar magnesium chloride and 2 micromolar primer concentration. No amplification occurred with 1 millimolar magnesium chloride and 20 picomolar primer concentration. Low annealing temperatures and high primer concentration allow mispriming, whose product competes with the target sequence of primers.
By adjusting parameters like magnesium ion concentration, annealing temperatures, primer concentration, and template concentration, very sharp and intense bands of PCR amplification were obtained at their respective product sizes. This indicated that all these parameters have effects on activity of Taq DNA polymerases, which requires magnesium ions for its activity. The annealing temperatures ranging between 20 to 85 degrees Celsius gave best amplification results because enzyme activity is greatest between these two orders of magnitude. The concentration of Taq polymerase was also very important if its concentration is very high, or else non-specific background products may accumulate and insufficient desired products may be obtained if it's in low amount.
For the results, detection limits of PCR or sensitivity of PCR are as follows. Luria burtani agar, or LBA, contained the samples from a spiked water with a known microbial content and is diluted from a preferred 10 raised to negative 6 to 10 raised to negative 10 dilution, compared to 10 to 10 raised to negative 5, in which reading colony forming units is too difficult to count due to the numerous colony to total. PCR detection limit of Escherichia coli was determined to be low as 3 colony forming unit per ml. The two figures show the agarose gel detection sensitivity of PCR by sensitivity bands at various colony forming unit per ml, resulting from the spiked water samples. Cell densities affects the band intensities of each homologous PCR product, while enhancing enrichment time from 4 to 6 or 8 hours can also increase the PCR sensitivity.
The result for the protocol sample analysis as follows. The molecular methods have lower detection limits, hence, a preliminary enrichment step before the amplification is necessary. If in a water sample has as low as 1 to 3 colony forming unit per ml of E. coli, it may cause serious health risks to human beings. Next, the sample filtration step is coupled with enrichment because the real water samples have very low amount of pathogenic microbes and can be captured on the membrane filters for harvesting. This step did not interfere with the results because the products of PCR were obtained and gave sharp bands compared to the analysis without filtration during sensitivity detection.
And that concludes our presentation. We hope you understand and learn something regarding the development of PCR protocol for the detection of E. coli in drinking water that we presented. Thank you and have a good day.